exterior peripheral region. So thus it has a structure, interior structure is called backbone and the exterior structure is peripheral reactive center or reactive region. Okay, and this globular and fiber structure is organized in four different levels: primary, secondary, tertiary, quaternary. So primary structure is the one what we have seen just now, a linear polypeptide chain. So linear polypeptide chain made up of particular set of amino acid order. These secondary structures are a given objective to develop a tertiary structure which may be globular or fibrous, isn't it? How they form this globular structure in a concise manner from a fibrous chain of Nagana by forming another intermediate structure called secondary structures, which are otherwise known as motifs or super secondary structures in the final stage. So these secondary structures are created or generated from the polypeptide chain in three forms. One is alpha helix, another one beta sheet, another one is a beta loop. So based on the number of amino acids organized together among themselves by creating a hydrogen bond, different sort of secondary structures are created. If four amino acids together they interact among themselves and create a bond between themselves, it forms a helical structure. First four amino acids and the next four amino acids and next four amino acids. So likewise. So it will go on until one particular distance. If more than 5 or 6 amino acids, basically 10 amino acids or more, if they together form a kind of chain structure which is stabilized by internal hydrogen bonds. Okay, it looks like a sheet, isn't it? That's why it is called beta sheet. Okay, on the other hand, if shorter through 2 to 4 amino acids, they together form one small single curvilinear structure, that's what we call beta loops. So these beta loops are internodal connective structures between two different or two same secondary structures. So these secondary structures together they form one super secondary structure called motif kanna, and this motif in turn they are folded further and it forms a globular structure in which water liking hydrophilic regions are placed outside, water hating hydrophobic regions are placed inside, and these hydrophobic regions along with their secondary structure they form a backbone, so which is surrounded by water loving or a reactive hydrophilic outer or peripheral secondary structures. So this compact structure is what we say tertiary structure, and technically it is otherwise known domain. So domain is a functional architecture of a protein which will be having one region called active site. So this active site would be responsible for executing particular function, and in some cases according to the need these domain tertiary structures they bind among self and they create larger complex structures called quaternary structures. So as you would have seen in hemoglobin, where you would have seen four different domains. They are integrated together to form one bigger hemoglobin structure, where four oxygen atoms can be accommodated with the help of 4-5 ring. Okay, so let us not go into the detail in those technical things, which will be a deviation from our topic. Okay, so this is all about protein structure. So this is what you would have studied in textbooks too. A protein chain, they form a secondary structure, and these secondary structures are further folded and they are given a proper structure. It may be either globular or linear in shape. Okay, and this structure in turn, if required, they may produce a bit more larger macromolecular complex called quaternary structure. Okay, so this knowledge, okay, so or this process, protein structure folding, is going to be the basis for protein structure prediction. Okay, so that's what we are going to see in this third question. How? Okay, so in how we are going to see the principles behind protein structure prediction and strategies, then finally algorithms or methodologies or methods. Fine. So let us start with principles then. So basically two principles are fundamental in protein structure prediction process. One is conformational structure. Another one, energetics. Conformational. So what do you mean by conformational? Giving a defined or definite look to something is what you say conformational structure, isn't it? A protein as a chain, okay, so it is given a specific definite structure. Okay, so some part of a protein forms alpha helix, some part of your protein forms beta sheet. So how this definite conformational structures are emerging, and there should be a universal law, isn't it? So that's what we mean conformational structure. If you know that law, you will be able to imply it for any new protein, and the structure can be predicted or deciphered. That is the first principle. Second principle, energetics. To explain the concept of energetics, I would like to take an example of a human life in which a person would be working until 50 or 60 years to earn money and to save money, and with the money the person will be trying to enjoy his or her post retirement life in a casual manner. So until the retirement age, they will be working like anything by taking a lot of nutrient source, by taking a lot of energy from outside environment, and after retirement they would try to conserve their money energy by undergoing a kind of passive hibernation, in which they'll be taking rest in home, or and they'll be doing some leisure activity. So here actually the availability of energy determines the human activity, isn't it? Likewise, a protein is attaining its structure according to the energy level available in the surrounding environment and within itself. So that is what we will be studying in this concept called energetics. So with these two principles, we are going to study, or we are going to predict the protein structure. Fine. So let us start with conformational structure. So in conformational structure, we will be studying certain technical terms. Okay. So first thing, blocks. Second one, prints. Third one, motif. Fourth one, protein family. So blocks are a defined zone present in a polypeptide chain. So what are the defined zones? On what criteria they have been divided into different zones. Apna. So here we are taking secondary structures as a template. If or another thing is there, I forgot to mention. Topological constraints. So these two are the factors that determine the category blocks. Okay, so based on the types of secondary structures that are created by different regions in a protein, you will be able to categorize them into different zones. Okay, so for example, I am dividing it into 5 zones. So number 1 zone may be known to produce alpha helix, 1 or 2 or more alpha helixes, and there will be intermittent beta loops in found in between the alpha helixes, and the second zone may be forming beta sheet, and again third zone may be forming alpha helix. Fourth zone again it would form a alpha helix with the intermittent presence of beta sheets. It may be a mixture of both alpha and beta sheet. So based on the secondary structure, we are actually categorizing this protein chain into different blocks. Okay, so these blocks, they form a secondary structure, right? So how they are being formed, this region is forming this type of secondary structure. Okay, so how do we confirm this, Arnagana? So that is where the principle comes. First one, amino acid composition. Okay, so there are certain amino acids which should be mandatorily found in particular secondary structure, and there are some amino acids which will not be present in certain secondary structures. For example, alpha helix. In alpha helix, you will not find glycine. Glycine is called the alpha helical breaker. So because of the flexibility and because of its linear structural nature, the glycine is not preferred in the curvilinear helical structure formation. So this is the reason it is called helical breaker. Okay, glycine இருந்துச்சுன்னா அந்த இடத்துல helix form ஆகாது, it would be broken அப்படிங்கிறது தான், that's a general connotation. So if you are spotting the presence of glycine somewhere here in this region, okay, so that region should not be a part of loop. Loop இல்லாமல் if you are finding in any other place the presence of glycine, you can say that region will not form alpha helix. It may be a part of a loop, or it may be a part of beta sheet, or it may not form any part in a secondary structure. Okay, so likewise there are certain amino acids. Okay, so presence of branched amino acids. Okay, lysine, arginine. Okay, these are branched amino acids. So these branched amino acids will not be found in alpha helix. So only small aliphatic amino acids are found in general. Okay, so with this you can say a presence of aliphatic amino acids which may form a kind of curvilinear structure, that zone will form alpha helix. Okay, in the same logic, beta sheets, so the presence of glycine, okay, and the presence of branched amino acids together, they both are together, it means it may form a secondary structure called beta sheet. Okay, so if you are finding the presence of certain positively charged amino acid or negatively charged amino acids ordered in a sequential manner, that indicates that region will form beta sheet. Okay, so these are small logics with which you can predict whether a particular part of a protein chain will form alpha helix, beta sheet, or beta tin. For example, the third one, beta loop, presence of proline indicates there should be a possibility, high probability of beta loop structure formation, because if a chain is growing like this, A presence of proline will turn the direction into complete 180 degree. Okay, so proline is called cyclic amino acid. Okay, so whose structure would be like this. Okay, if one amino acid is interacting with the proline, okay, so next amino acid in general, in a normal condition, the next amino acid must come somewhere here or here. Okay, but it will not be happening. So this proline will bring the next amino acid exactly parallel to the first chain. So it will virtually make a hairpin bend in the structure. So that's why the presence of proline indicates the probability of beta loop formation in that particular structure. So with this idea, you can predict the secondary structures. To confirm whether this rule, okay, implied secondary structures are formed theoretically also, for that you will be using a concept called topological constant. In topological constant, you will be studying two things. One is hydrophilic, hydrophobic property, and next one, Ramachandran plot. So just now I told you presence of certain amino acids indicates the secondary structure formation. Likewise, the composition of amino acid would help you to understand whether the particular region in the protein chain will form the outer region or inner backbone regions. Okay, so if that amino acid is hydrophilic in nature or charged in nature, so either positive or negative charge in nature, they form secondary structures, and those secondary structures will become a part of outer regions in a protein. Okay, on the other hand, so there are amino acids, okay, which are water hating or not reactive to the water. So those amino acids, okay, may be a part of particular secondary structure. It may be alpha helix or beta sheet. So those secondary structure would definitely be placed inside the interior part of the protein. Okay, and with this another concept must be taken here, that is active site. Okay, so this active site is the concept I mentioned earlier, a functional part of a protein. Okay, so which is made up of a few amino acid combination. Okay, and these active sites are partially formed by both hydrophilic and hydrophilic amino acids. So if you are finding the presence of combination of hydrophilic and hydrophobic amino acids together in some cases as a fusion, okay, so there is a probability that region will form active site. Okay, if you want to confirm this, okay, there is another rule is there. If you are finding the presence of certain amino acids like arginine, lysine, histidine, asparagine, okay, in that hydrophobic hydrophilic fusion, that indicates that particular part of the protein is definitely the active site, and it will be playing a role in executing the function. So with this, okay, you will be coming to a conclusion whether the region you are looking, okay, comes at the outer or interior part of the protein. So one aspect will be clear. Okay, so next aspect is Ramachandran